Welcome to Science and Social Tube channel. Kindly do not forget to subscribe our channel by hitting the below subscribe button and press the bell icon.
But it is not like that. It is in fisheries, we have shellfishes, we have aquatic plants. Whatever you will get in the water, that comes under fisheries. As per the FEO, fisheries is the capture of wild fishes from water resources. What are the different types of water resources are available while we are getting the fish, or raising the fishes through the aquaculture. The culture practice what we are doing through the aquaculture, whether it's a marine culture, whether it's an inland culture or freshwater culture or freshwater culture, that all includes in fisheries. Fisheries has been mainly categorized into three types: inland fisheries, marine fisheries, and fisheries through aquaculture. So in the inland fisheries, there is a freshwater resources which are coming under inland fisheries, like rivers, reservoirs, lakes. We have a lot of resources in the form of the reservoirs, lakes, rivers in Kashmir, also in India also. We have here the cold water resources that comes under land fisheries. In the marine fisheries, as you know, there is the oceans, estuaries. In aquaculture, includes both, whether the culture is the marine or it's a freshwater, both fishes and the shellfishes. So these are the categories in which fisheries has been divided. So if you look about the statics, fisheries data, the total production in 2018 was 178.5 million metric tons, in which capture fisheries is more, as it's clearly visible there, it's 96.4, and aquaculture was 82.1. So per capita consumption, if we see around the world, it's 20. It's highest in Japan, but in India, it's near about 11 kg. If we further distribute it, dissect it into capture fisheries and this aquaculture productions, marine fisheries, you can see marine capture is more, it's near about 84, and inland capture in which water resource, freshwater resource, even colder water is including in that, it's just 12. So total the capture fisheries is more as compared to aquaculture. It's clearly visible, it is 96.4. But if you see the aquaculture, inland aquaculture contribution is more, it's near about 51 million metric tons. Marine is very less. Marine aquaculture is still growing, still developing. But total, if we see, it is 82 million metric tons. If we see in this graph, as per the FEO, Sophia, if you see in the capture fisheries, land capture fisheries, okay, fishes which are getting from the natural water resources, it's very less. If you see clearly, capture fisheries in the marine water, fishes which we are getting from the marine resources, it's huge. There is craft and gears, or there are personers, trawlers, marine marine ocean area is very vast. So a lot of catch, lot of fish resources, a lot of fish productions come from this marine capture. If you see the aquaculture, inland aquaculture, it is going continuously. It's surging. It's increasing day by day. But if you see the aquaculture in the marine sector, it's less. Its contribution is not that much. So if we see this per capita consumption fish utilizations, if we see the population and per capita consumption, it's increasing. As I told in the earlier slide, it's near about 20. So it's day by day, it's increasing. In some countries, it's very high. In countries like India, where people are mostly the non-veg, so it's very less. In Kashmir, also day by day, this fish consumption is increasing. If we see the top countries in the world which are producing the fishes, China is in the top, near about 15%. It contributes to total fish production of the world. It's then Peru, Indonesia, Russia, and the US. India is on the 6th number. It contributes near about 4% of fish production in the world. Is I am audible? Hello? Hello? Is I am audible?
Yes, sir, very much.
Okay, thank you. So if you look the resources, if you look, it was but I was talking about the fisheries. It was overall overview of the world, what is going on, what is the total production in land, marine capture, and other things, and per consumption. But if you look in Indian perspective, Indian resources, the total production in India in 2017-18 was near about 12 million metric tons. Second largest global producer is India in aquaculture. It has also the second position. in aquaculture production, near about 7.2 million tons. Freshwater is more as compared to freshwater. Aquaculture growth it's very high. It's growing very high as compared to other agriculture and allied sectors. Its growth is near about 6 to 7%. And in terms of values, if you see the export, it's near about 45 crores every year. Indian government's earning from the export. It's contributing more in the GDP. More than 1% GDP is contributing to the aquaculture export. So if you see the total production graph from 1950s to near about 2016-17, the inland production, if you see, inland production is continuously increasing. Our overall total production is also continuously increasing. And marine, due to the overexploitation, a lot of problems is with the marine sectors. That's why it is decreasing. It's not increasing. For aquaculture, people are going for aquaculture productions, from whether it is a shrimp production, whether it's Indian major carps, which is in India, or in Kashmir, rainbow trout. So aquaculture has a huge potential in the fishery sector, where people can develop entrepreneurship, people can earn money, people can generate revenue. People in shrimp industry, people are getting revenue in the form of the dollars. So a lot of potential, huge potential is in the aquaculture sector. So if we see the resources, Indian resources are tremendous. There is a lot of resources you can see. Particularly, you can look into this reservoirs. 3.1 million hectares in Kashmir. Also, we have the lakhs reservoirs. Huge resources are there. But if we talk about Kashmir, potential is very less. Production is very less here. So a lot of scope is there for the fisheries, or in India also, to increase aquaculture production. Because we have huge resources, whether it is in pharma ponds and tanks, 2.36 million hectares, or reservoirs, or rivers. But at the same time, if we see the biodiversity of the fishes in Indian context, if we talk, there are some marine fishes, freshwater fishes. Totally, it's near about 3,035, and there are some exotic fishes which is culturing in India and abroad also. So if we talk, what are the main constraints, main problems with aquaculture? Why, if we have a lot of resources, we have potential, why it's not growing, why the sector is not developing? The first and foremost constraint, what I have found out, it is a quality seed production. Both, I can talk about quality and quantity. We have just 40 million fries, 40 million seeds we are producing every year in India, but we need more than 100 million fries. So we have to look this area to both to quality and quantity. We have to increase this seed production. Second constraint that faces the aquaculture is the feed technology and the feed management. If you look in the fisheries which are culturing, the problem is that we don't have the feed. We don't have this different stages, larval feed or fry or fingerling feeds of the most of the fishes. In Kashmir also, we have the problem with this brown trout. We don't have the feed, larval feed particularly. So feed management is also the constraint, or also creating the problem. There are a lot of fish which are culturable, which we can culture, but we don't have the feed in the captive conditions to give to larva, to brood stock feeds. That aspect we have to also look in that. The another aspect is the disease diagnostic, surveillance, and management. Disease problem is everywhere. It is in aquaculture, in the shrimp industry. Earlier in the 90s, there was this WSSV, huge loss. Still, this disease doesn't have any control. A lot of other finfish fishes have also other diseases, which if once it comes in the pond, we cannot do anything. So that aspect is also creating problem to the aquaculture development. Other important aspects, which I think was the constraint, was vertical expansion, species and system diversification. If we look in the Indian scenario, if I cannot talk about the China, where more than 100 or 150 fishes they have aquaculture techniques, breeding techniques. But in India, there are few fishes which we are culturing, whether it's a rohu, katla, IMCs. In itself in Kashmir also, we are only culturing this rainbow trout. No other diversification, no other fishes aquaculture technology or aquaculture system or breeding technology is standardized or is available for the farmers, so that we can go for aquaculture system or aquaculture. So another issue is the post-harvesting. As you know, fish is the highly perishable. Okay, fish is very highly perishable. If we cannot take the care during the post-harvesting, within 1 hour, within 2 hours, it will be gone. Nothing will be there. So access, another one I thought is access to technology at the grassroots level. Still in India also, people are doing some extensive type aquaculture system. Traditional way of doing the aquaculture system, they doesn't have the technology. We have to go there. We have to show them what are the different techniques we can use to develop this aquaculture. What are the recent techniques? whether it's a biofloc, whether it's a respiratory system. So that techniques need to be reached to the farmers. And another important aspect that nowadays people are talking so much about, that's the climate change. Climate change is also affecting the fishes, whether it's with respect to thermal temperature change or with respect to salinity and other things. So these are the problems that I have found out. These are the issues which hindrance, which creates the problem for the aquaculture development in world, also in India particularly, and in Kashmir also we have these problems. So in order to eradicate these problems, in order to tackle with these problems, there are a lot of platforms which are nowadays available for the scientists, from the researchers, that they can use. This first and foremost is the genome editing methods. So if we go to molecular aspects, like whether it's a transgenic fish, whether it's a recent CRISPR case, that techniques we can use to grow the fish, to manipulate their genome, to try to find out their which genes are responsible for the growth. We can play with them, we can fast their growth rate. That approach we have available, people are using that. There is one GMO which is available with the US people, that's aquabound salmon, that has more growth rate as compared to the wild salmon. There's another approach that we can use for developing for increasing the production and productivity of aquaculture, is the genetic approach. We can go different approach of genetic, like selective breeding programs are there. We can go for the selective breeding so that we can develop some good strains that will show the best growth, that will show the more growth rate. And another recent aspect is the artificial intelligence. We can try the different artificial intelligence or machine learning techniques that will be helpful for aquaculture. Another area which is nowadays also growing, like anything like computational biology, we have a lot of NGS platform. We have a lot of techniques which are available. By using different bioinformatics software, we can get a lot of we can take a lot of ways, a lot of pathways, a lot of cellular and molecular functions. So another things that recently people are using in aquaculture, the techniques which people are using for increasing the production, whether it is a cellular system or biofloc system. And another, this is cage culture. Cage culture is also growing. Some of the fish are showing good production, like in tilapia or pangases in India. They are doing this cage culture for increasing the productions. So another platform that's available nowadays for the scientists, that today the talk is about, is the nanotechnology and the nanoscience. So I cannot talk about the other aspects where we have used this in the fisheries, whether it's the genome or genetics or artificial intelligence or computational biology. So this talk is on this nanotechnology and nanoscience. I'm going to talk about this nanoscience. So as for the last 10 days, you people are learning so many things in the nanotechnology. Some aspects is the same as people. This nanotechnology has got the attention from, I think, 2000 last from 2 decades. People was crazy about this technology, particularly this electronics and medical science. People are exploiting it like anything. This nanotechnology, as per this and Nanotechnology National Institute US, they define it understanding and control the metals in the dimension of roughly 1 to 100 nanometers. They are telling this Nanotechnology National Institute, the definition of the nanoparticles or the technology, the nanotechnology is dealing with the substances that range in the size of 1 to 100 nanometers. But some assumptions are there. Some people are talking about higher size also. We will discuss this later on. Yeah, in the last 10 days also, you maybe have learned so many things, so many people. I was himself listing the lectures. There was some this debate on this, how much size should be there, and this one and that one. The term this nanotechnology has been used by this scientist in 1974. So the person who is behind this nanotechnology, as you people have seen, so many people are talking about this slide. Also, this Dr. Sir Richard Feynman, who is known as father of nanotechnology, in 1959, he gave the lecture to students. Oh, at the bottom level, at the atomic level, we have new kind of forces. There are new kinds of possibilities. We can manipulate them. We can play with them. Dr. Richard Feynman, sir, he was a theoretical physics person. He works on the quantum physics. So when he sees that there are different types of forces, different type of possibilities, different types of things that we can change at the bottom level, then nanotechnology slowly start developing. And I think in 1965, he got the Nobel Prize in the quantum physics. So if we compare the scale of nanoscale objects, natural things, what we have, like we have the DNA, okay, that's 2 nanometers width. We have these proteins, okay, their size is there, and we have the artificially created things, like you have the fluorines, it's a carbon form, its size is there 1 nanometer. We have the nanotubes that we have with these quantum dots. They have people are exporting like anything, these quantum dots. They have specific property of optical properties. These dendrimers, they are again polymeric. This nanoparticle, their size is near about 100 to they are about 10 nanometers. Yeah, you will see the virus size. Here the slides aim is to compare what are the nature things are available with respect to the people they have manufactured. So you can imagine, you can see how this nanotechnology or nanoscience is important. So we have already in the biological system, we have the biomolecules, we have chemicals which are in the nanoscience range. So the question comes, why nanoparticles are interesting? Why people are behind this nanotechnology? The answer is these properties, what you are saying at the left side of my mind, whether it's catalytic property, electrical property, magnetic, mechanical, biological, that we people, agriculture, applied people or medical science people are interested in. The biological properties, these nanoparticles show, it's tremendous. It is incredible. It's totally incredible. You will see it's increasing permeability through biological membranes. Okay, it's not easy to pass a biological membrane, but these nanoparticles, which are in size of 1 to 100 nanometer, they can easily pass to this biological membranes. That property that scientists have used nowadays, they are playing with that. They are using that with different applications. Yeah. Another property, that important property with respect to nano scales or nanoparticles, is the catalytic property. Better catalytic efficiency through high surface to volume ratio. That is the important property that nanoparticles. So it has more. It has high this surface to volume ratio. Other properties, it has more electroconductivity, optical, that we as agriculture scientists, we don't have that much concern, but we are more about this biological properties. So if you see the significance, why people are synthesizing nanoparticles, whether they are using organic or inorganic polymers, or they are using some metals or inorganic problems, why these nanoparticles, what is their significance? First and foremost is the nanoparticles have generally higher intracellular uptake compared to the microparticles. The cells, the biological system, in the cell membrane or in the intracellular cavities, these are easily passing. These are easily passing the cell membranes as compared to higher micromolecular size. Researchers have already people have find out that 100 nano-sized nanoparticles shows 2.4-fold greater uptake as compared to 1 micrometers microparticles. So another thing that people are interested, and a lot of talks was on that aspects also in this training, that's provide a target delivery. Yeah, you are talking about this term, the smart drug delivery. So you have this sustained release, or you have the particular, you are encapsulating or you are entrapping some specific drugs in the particles, and you are releasing at a particular target, so that there will be the less toxicity effect. There will be more bioavailability to that particular organ or for that treatment. Another aspect that we people have worked out, that we people was interested more, or this nutrition people may be more interested to improve the stability of therapeutic agents against enzyme degradation. As you know, in our body, in the biological system, there are a lot of enzymes which degrades the substance, whether it's a proteinase which acts on the proteins, whether nucleases, they are degrading RNAs, DNAs. So to increase the stability, we are coating we are encapsulating them in some polymers or some encapsulations in order to protect, or in order to make the more bioavailability to available to that particular organ or to that particular treatment. So these are some significance that people are interested, or people have shown interest to work on the nanoparticles, because these are the significance that nanoparticles have shown as compared to the higher particles. So we have, as you maybe have seen this slide also for so many presentations earlier. Also, there are different types of nanoparticles which are available, whether they are from the organic background, like this copper. Yesterday I have seen the lecture on the copper nanoparticles was there. People was talking, there is a silver, there is a gold, there are polymeric nanoparticles. I have personal experience with this polymer nanoparticles. I have worked on the chitin sodium alginates we have worked on. So I'm going to talk about that polymer nanoparticles later on. So we have available there by the liposomes, their dendrimers, their nanotubes, their nanocrystals there. In the market, you can get this inorganic nanoparticles from the companies ready-made, whatever size you want as per your experiment. You can this order from Sigma or some other, whether you want gold, 5 nanometers or 20 nanometers, whatever you want, you can get ready-made, or you can synthesize in a lab. Also, there are the protocols which are available in the literature. You can go through what are the different methods we can use for synthesis of both this polymeric and the inorganic nanoparticles. So, as I told, I have my personal experience to work on this polymer nanoparticles. Let's talk about polymers. So, if we talk about the classification of polymers, There are natural polymers, there are synthetic polymers. You may be knowing there are semi-synthetic polymers. If we talk about the natural polymers, you have proteins. Proteins are polymers made of amino acids, peptides. So there's a rubber, there are synthetic polymers, polythene, there are semi-synthetic polymers like rayon. So we are talking here, I will talk about the natural polymer. Before talking about the natural polymer, we should we should know what is the criteria for ideal polymeric clear nanoparticles. If you are selecting some nanoparticles for the drug delivery or for some other purpose for DNA vaccines, what we have worked, I will talk about later on the DNA vaccines. So you should know that polymer should be easy synthesized. There should not be complicated with respect to its characterization or synthesis. It should be biodegradable. That's very important property. It should not be the non-biodegradable. If you would go in the biological system, if it will not show any effect, but it should not be the toxic also. Okay, it should be biodegradable. It should be degradable. It should be biocompatible with the living system, living tissues. It should not be the this toxic or important. Another important thing is it should be inexpensive. It should not be that much costly that hire only a lot of funding needs for the purchasing this polymer. So these are the properties, or these are this criteria which you should keep in mind when you are selecting some polymers for the nanotechnology or for nanoparticle synthesis. Here I am talking about the chitosan. I have personal experience with our group has work. We have done a lot of work on this chitosan, and I can proudly say this chitosan is our own product. It's a fisheries product. We are extracting it from the chitin, and this chitin is available with these shells, whatever these invertebrates are there, invertebrate shells, there with crab, prawns. It is extracted from there, from chitin, by doing the distillations. Okay, we are removing this style group from the chitin. And we are getting this chitosan. You can see here, there are the two structures which is available here. This chitin, which I told you we are getting from. It's a fisheries product. It has a lot of applications. It's a millennium product. It has tremendous applications everywhere in every sector in the field, including agricultures. So these are getting from mostly from the invertebrates, whether from the crab shell or this prawn shells. So removing it is this style group. We are getting this chitosan, and that chitosan is people are using for a lot of applications. I have bring here these applications of the chitosan later on. I will talk about that. But here, this chitosan, if we look into this chitosan, it's the second most abundant natural polymer after the cellulose, which is found in the plants. That's the most abundant polymer which is available for us. But this chitosan is the second most polymer. It's, as I told you, it's available with the crustaceans or this crabs, marine shrimps, freshwater shrimps. The important characteristic that when I have also prepared myself this chitosan nanoparticles, it is not dissolved in the aqueous solution. You should have you should give it some acidic conditions near about pH between 5.5 to 6. Then it's dissolved in the aquatic condition. It's soluble in dilute acids. That it's already written there. It's insoluble in the aqueous solution, but you have to give some acidic condition so that it will dissolve. When we was preparing the nanoparticles, we give the acetic acids. Okay, we maintain the pH between 5.5 to 6, and so it became easily dissolved in the aqueous solution. Another important property this chitosan has a positive charge. Okay, it's a polysaccharide, the positive charge. So when it is a positive charge, that means you can easily bind it with any drug. Okay, and with DNA. Okay, DNA, you know, it's a negative charge. Amino acids, those amino acids which are negative charge, you can easily bind with it. You can easily conjugate with it, and it has a property that I already told you. It's a it's a biodegradable, it's a biocompatible, it's an organic substance. It's not inorganic. Okay, it has a reactive to groups. It has this style group through which it can interact, or it can react with some other drugs also. So I will talk about these chitosans. We have worked on this. Just I have the application of this chitin and chitosan. You can see everywhere, whether it's water treatment, it's agriculture, biotechnology, okay, food and supplements. Okay, it's used well known in fisheries also. It's using as a blank chitosan. I'm talking about a nanotechnology or nanoparticles. It's using as blank aminostructure. Okay, aminostructure given as aminostructure. If you see in the biomedical applications, you can see here there's a drug delivery, gene delivery. Okay, so many things are there in food. Food and health supplements. You can see food additives, including this filtrations, papers and films, fertilizer, agriculture, for the agriculture, antimicrobial agents. Okay, so this chitin and chitosan is known as the millennium product. It has application like incredible. It's incredible product. So talking about This nano revolution, as our talk is on that, on the nanotechnology. If we see the nano revolutions, if we talk about the revolution, the first revolution which starts in the world is the industrial revolution. It was earlier in 1700s. Okay, that industry setup and industrial things has been come in the world. Then the revolution of electrification, electronics things has been come forward. So many electronic things has been come. Then revolution in information technology and this IT sector, this computer science has been started in the third revolution. But after 2000, that revolution of nanotechnology has been started. People have started working on the nanotechnology. Whether you have seen this electronic, the small chips are there. Okay, nano chips are there. So many things have come now. Everywhere in our life, nanotechnology is there because everything is connected with the nanotechnology. It is a faster, thinner, tinier, and more practical. So we, from 2000 onwards, we are living in the revolution of the nanotechnology. Nanotechnology can get everything are coming in the nano size. Here again, in these slides, I have shown which development has been taken place with respect to nanotechnology when we are talking about the revolution of nanotechnology has started from 2000 onwards. So people were in the first generation or first product which has come, they started since the nanoparticles, coating of particles, nanomaterials. These things has been coming, and slowly as from 2015 to 2020 onwards, a lot of things has come in this nanotechnology. That nanopore sequencing is there, that NGS platform is there. Yeah, these 3D structures are coming with respect to nanotechnology. So it's building this sector. This revolution of nanotechnology is continuously going like anything. It's showing tremendous application everywhere, whether it's electronics, medical science, agriculture, or some other sectors. So if you see the response of a market of these nano products that manufacturer companies has shown, it has been remarkable. It's incredible. If you see earlier during the earlier stage, it was just 25%, then gradually increase from 2015 onwards. The market, most of the products which are coming in something here and there, whether it's a drug, whether it's electronics, whether it's in some agriculture products also. Sure, something nano-related things are there, nano products are there. So this companies, this commercial companies also shown tremendous response for manufacturing this nanoparticles. There is a lot of scope still for agriculture and allied products. So many products are not available for agriculture and or veterinary or fisheries products. We have a lot of scopes in this area. So coming to the main topic that I was interested to talk, that was assigned to me to talk about. What's the application of this nanotechnology in the fisheries and aquaculture? So if you see, I have bring this diagram, utilization of nanotechnology in aquaculture and fisheries. That application, if you see the smart drug delivery and nano vaccine development, that himself I have worked on these aspects. I will talk about my work things. Yeah, feed or nutraceutical delivery. As I told earlier, this nanoparticles have this applications. They will protect your feed or nutraceuticals, whether it's vitamin C or which is something else from proteases for nucleases. That's why the nano nutraceutical delivery or nano delivery was important with respect to the feed. Gene delivery. Okay, we have himself tried the gene delivery with respect to different disease. Okay, we have construct some DNA constraints to deliver with this, or another things that we have also work on this. There's a diagnosis of fish pathogens. There are some kits are available in which golden nanoparticles are there. You can easily differentiate these pathogens. That kits are nowadays available. As I was earlier talk about this nanopore sequencing of whole genome sequencings are available with the nanopore sequence by nanotechnology has been used. This tagging and barcoding is there, or water filtration and remediation. This is also important aspect as far as aquaculture is concerned. We have a lot of algal bloom problems. People have people have come out with some commercial products also with respect to water filtration and remediations. Let's talk one by one all these aspects. I can show my work also and what's going on around the world with respect to nanotechnology. So this is the paper that's my work during my master's work. We have published this paper in PLOS. So our aim was in the fisheries, in animal science also, people are using hormones for the breeding purpose. But in the case of the fishes, we have big problems in a lot of fishes where females are maturing late, males maturing earlier. That was the problem. Yeah, females are maturing, but they're not releasing the eggs. There was a problem with the reproductive output also. What we thought, okay, we will use this nanotechnology. We have used the hormone, this leucogenic hormone, releasing hormone. Okay, we have used chitosan particles, nanoparticles, chitosan. We have synthesized, we have conjugated with LHRH hormone, and we have also used chitosan gold LHRH. We have used both organic and inorganic to see what will be the effect on the reproductive output of this fish. So we have synthesized first this gold nanoparticles, okay, with the chitosan. We have changed the concentration of this chitosan, but gold concentration we have keep seeing. You can see with respect to concentration, different coloration, it's showing this gold and chitosan solutions. Now, here in this diagram, what our concern was, how we will interact it, how we will encapsulate this LHRH. So what we have found out, we first tried to react this chitosan with this gold nanoparticles. We have given the heat, okay. So this gold was got inside, and this chitosan got outside. Then we put the hormone, that's LHRHs, and it get entrapped with this complex, this gold and chitosan complex. So we use this formula. We set our experiment. This was the distribution of this nanoparticles by using this TLS nanoparticle analyzer, and there was a scanning electron microscope image also. The distribution, you can see this machine, the nano analyzer is like a normal distribution where you are getting the peak. Sometimes you will get the 2 peak. We will see in some results also later on in my presentations. So this was the distribution. It is just a normal distribution, and we are at the center. You will take the average how much nanoparticle size is there. So after injection to the fish, we was interested to find out serum hormones, what will happen to the hormone profile. Okay, we have injected this hormone. We have injected naked bare hormones in which there was no nothing nanotechnology, the LHRH, and we have used this chitosan and nanoparticle LHRH. We have used chitosan gold LHR nanoparticles, and we have used the control. We were surprised to see there was a sustained release. There was sustained release with respect to this chitosan nanoparticles with gold and chitosan nanoparticle with LHRH. You can clearly see that there's an upsurge of these nanoparticles after the post harvest of 24 hours. So this was tremendous. This was incredible results to find out. No, hormones has been sustained. This LHRH is it's not degrading. Its half-life in the blood in this has been increasing. So that's why this sustain was continuously going on. So we have got this tremendous result. We thought, okay, this will be good to mature the fishes to see the reproductive output, to go for the reproductive output, to see the fishes which matures late, so that there will be sustained release of this LHRH hormones, so that gonadal development, so that ovaries will be developed, particularly with respect to females which have the more maturation or which have the more complicated reproductive system. So here is the reproductive output, the eggs which fish was releasing, the fish which was we use, it is the common cargogen in the experiment. You can see the reproductive output is more in chitosan nanoparticles and chitosan gold nanoparticles as compared to blank or as compared to the naked this LHR hormone. So this was hormonal profile and reproductive output was okay, but we was also interested to sound what will happen to the histopathology aspects. We take the ovary and we find out there was a release of the eggs. If you see in the last treatment, treatment where this chitosan particles, gold nanoparticles, chitosan gold LHR nanoparticles was used. So egg release was more, maximum egg release was from there as compared. You can see here we were seeing some mature oocytes are there. The eggs release is not that much as compared to you can see on CMD treatment. So this result was incredible. We was sure, okay, it is the formulation, it's the formula which has been worked out. We have whatever the hypothesis we was having, we need to sustain these hormones in the blood, in the blood of the animals, so that there will be more maturation. There will be this in the fishes. Otherwise, we have to give to multiple injections for the maturation or for the breeding purpose. So in this, we have give only the one injection. We do not give the multiple injection for the maturation or the breeding purpose. So another work which was my PhD work. During my PhD, I worked on the kiss-captain, which is the peptide, which is responsible for releasing this GnRH from this hypothalamus. So we characterized that gene and we synthesized the peptides. Okay, this peptide, which was a functional peptide, a functional domain of this kiss-captain was the histone. That was the decapeptide which was responsible for functional domain. So we try to encapsulate these peptides we extract from the fish. We synthesize it. Okay, we synthesize it. We nano encapsulate with it. Again, we have used the chitosan nanoparticles. This is decapeptide that I was talking. These 10 kiss amino acids which are functional responsible for this for initiating this GnRH release from the hypothalamus. When the GnRH is releasing automatically, that comes to the pituitary, interior pituitary, and so that LH and FSH, which are responsible for the maturation and this gonadal development that starts releasing. So we synthesize this kiss peptide. At the end, there was a tyrosine. You can see tyrosine. Okay, there was if you see the tyrosine, this functional groups. It was a carboxyl group at the end, and this is the chitosan formula. If you remember, I have seen also the conversion. So there was an interaction. We have used the magnetic stir for preparing this magnetic stir nanoparticles. So this was the nano conjugate complex, chitosan kiss peptide conjugate complex that we have formulated. So after we have gone for the characterizations, we have gotten this size and other properties like charge and polydispersity that determines whether it's a homogeneous solution or heterogeneous solution. That things we have found out, and we have found the interact efficiency also. It shows that 60%, 64%. This peptides, this peptides has been entrapted in the chitosan. So then after giving the injection to the fish, we analyze the hormones, LH and FSH in fisheries. We are talking sometimes GTH1 and GTH first. So again, there was a sustained release of this chitosan and nano encapsulated things. So we confirm back again. No, what I got the results in the masters. It was some nano encapsulation was showing some sustained release of these peptides of this drug. So we can increase the shelf life and we can use for the fishes which are maturing very late. That was the formulations that we have developed. So another area, another work that we have published was in Journal of Fish Disease. I was also the co-author of this. One of my this junior, she's present as scientist in I think in SIBA, Central Institute of Aquaculture. What were our intention? There is a freshwater prawn called Mycomacron rosenbergi, in which the nodovirus, okay, which causes a lot of diseases, necrosis, other diseases which are very harmful for this culture. We have taken the gene that's known as extra small virus antisense gene. Okay, we have conjugated that. We have developed the nanovaccine type. We have conjugated that gene. We have extracted that construct, antisense construct, and we have conjugated with that this chitosan. So these, when we have conjugated with that chitosan, and we set the experiments, there was these experiments were there through feed. We have used different methods in the fishes. We can give the dip treatment also. We can use through feed. Okay, we can go if the fish is big, we can go for the injection also. So these are the experiments that we have set up with respect to this Mycomacron rosenbergi. So, and this was the this AFMS atomic force microscope image, 3D image of these nanoparticles. So these were the experiments. The treatment, 5 treatment was there was positive control and negative control, and there was a treatment through feed and through immersion treatment with gene antisense gene and chitosan. So this was the persistence study given through different treatments, whether through feed, whether through immersion. You can see there is this by using the primers of that gene, we have amplified again, and we have found that gene's persistence there in the body of that prawn, and we have found also the survival rate was good with respect to this antisense chitosan nanoparticles. So this result was also good. We got survival, we got protection by using this antisense with chitosan nanoparticles. Another paper that was my work also. We tried to use this green synthesis. People are talking about the green synthesis nanoparticles. They are important. Here we have used this name with the silver, and we have found out what will be its response, because people are talking the silver and neem. It has also individually without nanotechnology using it shows it's antifungal or it is an immune response. It shows immune response. So we synthesized. We have done the synthesis of this green neem with the silver and nanoparticles, and the particle size was again analyzed. We was having there in CIFE under the sir whom I have done my master's and PhD. He was having the project on nanotechnology. He was having this nano analyzer that works on the basis of the dynamic light scattering phenomena, and we have analyzed the particle size and zeta potential and other properties which are necessary for characterization these particles. So these are the synthesis nanoparticles after 12 hours. This was the coloration. So people have check also with the UV wavelength and this was after 24 hours. So we checked the experiment and we have given, this was the distribution of this nanoparticle. Sometimes with this GLS, you will get the 2 peaks. Okay, you will get to 2 peaks, and it will give at the outside, it will give all the properties in separate page. It will give the average, what is the average size of the nanoparticles, and this is a zeta potential, which is a charge which is necessary to see whether its particles are stable or not. So this was the experiment after making this control and blank neem or silver with neem nanoparticles, and we have a challenge with Aromonas heterophylla bacteria which causes disease to fish. So we have find out the survival, and our treatments was showing the good response with respect to the survival percentage. Another my batchmate has worked where I was also the co-author was this paper which was published in the current Nanoscience. That's RNA loaded cytosol nanoparticles for enhancing. Again, we have used these nucleotides as I told earlier. These nucleotides, if we use DR or NAC nucleotides in the body, these DNAs or RNAs will attack on that. It will degrade it. So very less availability will be for this for the organ, for the animals. Because this RNA synthesis also RNA nucleotide synthesis also needs a lot of energy inside the body, but it shows the immune response. So we set that based upon that hypothesis, that consumption, we challenge again with Aromonas heterophylla disease with fish. So we use different concentration of this RNA and nanoparticles with 0.2%, 0.4%, and we have calculated hematological parameters or immune response after challenging fish with the Aromonas heterophylla. The fish was selected was the Lobo orohita. So this was the particle size and its charge or its entrapped efficiency, that's RNA entrapped with the chitosan. These are the physical chemical property which was necessary to estimate after conjugating this chitosan with RNA. So after doing all these things, we got for the hematological and this and immunological parameters. You can see here clearly in 0.4% RNA chitosan treatment, the specific growth rate is more. So it was showing the more growth rate as compared to other treatments, or its feed conversion FCR was more and protein efficiency was more, or this its visceral somatic index. Okay, so somatic index was also more with respect to this chitosan RNA nanoparticles. So as people are talking so many things with respect to this inorganic nanoparticles, even there are also some reports with respect to the organic nanoparticles, whether there is a toxicity with respect to these nanoparticles. One of my junior has done the research work where I was also the co-author. He works on the silver nanoparticles. Okay, we was trying to find out what will happen at the DNA. Okay, how will be its molecular response? So it was his master's work. It got published in this good journal, Food and Chemical Toxicology. What we did, we synthesized the nanoparticles of two sizes. Okay, here was our highlights was silver nanoparticle toxicity is a size and dose dependent. So we was trying to find out what actually which dose is suitable where it will not cause toxicity, and what is dose and size has some interrelation with each other, whether there is toxicity with related to that. What is the scenario that we was interested? Silver nanoparticle accumulation increases in the liver as the exposure time increases. That was also the thing. It accumulates silver nanoparticles, inorganic nanoparticles get accumulated as the time of exposure. The more you keep the exposure, it gets increased in the liver. Exposure of nanoparticles cause down regulation of genes. So we check some genes which are responsible for the stress genes, whether it's a heat shock protein, whether it's a methyl. We check gene expression. Also, we find out whether the expression has increased or decreased. That I will talk about later. But our intention was here we have used different concentration of the silver nanoparticles, but the size was two. We use 18 nanometers and 29 nanometers silver nanoparticles with respect to these concentration 100, 204, 800. So the graph in front of you is the DNA damage. Okay, percentage of DNA damage with respect to the days and size. There are only two sizes. If you see clearly, 29 and 29 and 18. If you see the DNA damage is more with respect to 29 nanometers at the 7th day. Okay, like that you can see the more higher the size or more higher concentration, the more damage is there. So this is the image of this comet assay. The first one, A, is the 29 nanometer. You can see the cells, the DNA is totally degraded. Okay, just dispersed. And another one, 18 one, it's not that much. 18 is not that much. If you see the positive control, which we have used as the DMSO, and it is a control cell that the dispersion control states it's compact, it's not a dispersal. So by using this comet assay, we can find out there is people are talking about so much about this inorganic nanoparticles, whether it's a gold, silver, toxicity, it shows toxicity. It is necessary. You have to evaluate that. You have to find out which size or which dose is suitable for the particular organisms. Toxicity is sure there with respect to the nanoparticles, whether it's in the environment or in in vivo system. So as I told earlier, we was also interested to check the gene expressions. You can clearly see the gene expression of by using the real time gene expression of heat shock proteins. Okay, you can see the expressions here are changing tremendously. You can see this macrothene. You can see the expressions of 1800. Okay, 1800 and this of 400. It is more with respect to exposure of the day as compared with both this 18 nanometer and 29 nanometer. So it was stress genes was responding. Stress genes were responding. That automatically also has been clarified by this comment. You know, there is a damage. There is a response at the molecular level which is changing. So it was our work that I was interested to share with. We have some other papers was also related with whether hydrogen or silver nanoparticles or gold. We have worked out, but it was not. I thought no, it's not necessary to talk about all my own work. So that's why I bring some other works which is going on around the world. So if we talk about water filtration and remediation, because we are dealing with aquaculture here, we are fisheries. When the fisheries, we are talking about the fisheries in the water. So people have used different types of nanoparticles for removing this, whether it's bacteria, whether it's nitrites, whether it's fluorides, whether it's toxic substance. So people try so many things, whether it's a silver, whether it's a gold, whether it's iron nanoparticles. People have reported there are a lot of reports which are available. No, it is showing effect. It is silver or gold or iron. It is effective against to remove these bacteria or to remove these contaminants. If we see here, this was another paper from somewhere. I didn't. Sorry, I forget to give the references. I don't know where I have taken from this, but the intention is that they have used this E. coli genome DNA. Okay, they have used this gold nanoparticles oligoprobes. You can probe. They have used. They hybridize each other, and if it is there, it is there. It gives the colorations. Okay, it will give the red colorations. That means it's a positive. That's E. coli DNA is there. If it is not there, it will give the purple color. So it was a simple test by using this gold with oligos probes to find out whether in the water or in the water sample there E. coli is there or not. So it is nanotechnology and test, very simple test is to find out. So what they was doing with this respect to this, to removing the harmful substance or harmful chemicals from the water. You can see here nanoparticles that they have formulated this hydrogel films, or they have absorbed it in some absorbent substances. And here you can see there is a water with different pollutants like nitrites, fluorides are there, or bacteria are there. And then after putting the nanoparticles, you can see a lot of near about 60-80% pollutions has been removed. That is the principle. That is the reason behind this nanoparticles. They are helping to remove this toxic substance or bacteria from the water. So this here is the image of this is a nano biosensor which has been developed by the NASA people to find out this salmonella in the water sample. So actually, if we see how these silver or gold or some other nanoparticles are functioning, how they are antimicrobial, how they are against these bacteria. Here you can see there are different types of nanoparticles. They have whether silver is a titanium, zinc, or magnesium. And they have find out what is the actual mechanism, how they are protecting it, whether they are releasing serine ions, damages proteins, okay, suppress the DNA replication of that particular bacteria or microorganisms. Okay, yeah, with respect to this magnesium nanoparticles, membrane damage, or this is zinc, release of zinc, production of hydrogen peroxide, membrane damage, production of oxygen. So these are the mechanisms through which these nanoparticles helps you to get rid from these harmful bacteria or get rid from these pollutants. There was a lot of work also done with respect to this DNA nanovaccines. Okay, one of the reports from, I think it's from China, this paper, what they have done, they have find out this Vibrio parahaemorrhagiticus, which causes disease. There's one fish called sea bream. They have used this gene called outer membrane gene, K gene, and encapsulated with, conjugated with this chitosan nanoparticles, and they have find out expression of this outer membrane gene within 3 weeks evokes immune response. So it shows immune response with respect to this Vibrio hamilticus, okay, and they have found out the survival rate of more than 70%, 80% survival after 3 post-week vaccinations with chitosan and plasmid DNA of that gene. So there was another report, a very good report from India also, Dr. Sahul Hamid sir, who has worked on this WSSA, which creates a huge problem in the chemical industry in the early 90s, as still is going on. Our Kashmiri students also working on this DNA nanovaccines. Very soon, I think they will go for the commercial products in CIFE. His PhD work is on that. So what Sahul Hamid sir's paper is also the oral derivative DNA construct. What they have done, they have taken this viral protein gene construct and they have conjugated it, they have constructed it with nanoparticles and then challenging with this virus WSSV and to find out this survival. So their results were also good. Their survival was after shrimp at 7, 15 or 30 days post-treatment, rate of survival was 80%, 60% respectively, whereas mortality was observed in the control group. So this was great work from India also. And also you can see that they have using the cell line, they have checked out whether this DNA construct is going inside the cell by using immunofluorescent techniques. Okay, and they have used CBUS cell line. They have developed their own cell lines. This SK cell line is Sahul Indian CBUS kidney cell lines. Okay, he has developed its own cell line from this CBUS. It was the first cell line from marine fish. So they have checked this expression of this construct inside the cell line also. It's clearly visible. You can see here clearly visible the expression of this DNA construct. So people have also tried this nutraceutical delivery that I was earlier talking about, the nutraceutical delivery. This paper was from Spain, where they have used the vitamin C. As you know, vitamin C is anti-stress. Okay, so they have found out this rotifers, which is an invertebrate, okay, which we are using as a food for many fishes. Many fish as a larva feed. So they have found out this rotifers in up to 2-fold of ascorbic level in comparison to control group. Okay, so people have tried this, nutraceuticals also. But what was the benefit of this nutraceuticals? It is in front of you. Okay, benefit of this derivative of the nutraceuticals, different nutrients, taste, mask, and protection from degradation during processing, enhanced bioavailability of nutrients, okay, antimicrobial health benefits. That was the benefits of delivering these nutraceuticals. And people have also tried these nanoparticles to find out whether it will influence the growth or not. Scientists from Russia, okay, they have reported there's a faster growth rate when they have fed the nanoparticles, this iron nanoparticles, nanoparticle in sturgeon. Sturgeon is the fish which is mostly cultured in Russia. They have found out there are about 30 to 40%, 30 to 24% growth rate has been increasing by using these nanoparticles. And from China, they have found out also the selenium. Okay, selenium supplements has also increased the growth rate in Caricis aureata, that's a crucian carp fish. So what actually happens with these nanoparticles happens easily pass through the microvilli of the intestines. It got easily absorbed, and that's why it's a faster absorption in the body, and the growth rate is more. Detection of pathogens, people have also tried this nanotechnology in fisheries. Also, they have used this golden nanoparticles and antibodies of that particular organisms, and find out this based upon this calorimetry or with other techniques like loop mediated isothermal. They have found out we can easily detect with high sensitivity and specificity these different types of viruses or bacteria which are harmful to the fishes. So another important thing that creates problem to the fisheries is in the ponds. You may have seen there is algal bloom, lot of blooms, lot of algae is creating a problem for the cultures, and through which everything is collapsing during the pond productivity. So by using this 14 nanometers lithium based nanoparticles, they have find out this eutrophication. Mostly this algal bloom is due to the eutrophication, due to the phosphate level which is present in the water. They have find out no, it is absorbing it is reducing this phosphate level and ultimately prevents the growth in algal growth. So there is a technology which has been used in this technology has been used in the rambotrope culture, particularly in the US, for cleaning this ponds for the algal blooms. So in the post-harvesting, as I told, the fish is highly perishable. Also, within 2 hours or 3 hours, it will be go on. Different types of coating materials, different type of coatings, phytosans or silica, some other materials has been used to maintain the temperature, to maintain the freshness, to maintain the integrity, or to protect it from the pathogens in post-harvest. Also, a lot of work has been done with respect to this nanotechnology that we are calling it the smart packaging. So there are some commercial products which are available with respect to aquaculture and fisheries, which we can use, which are available in the market, where the nanotechnology has been used, where the nano products has been utilized. First is the Nanocheck. Okay, it is used for estimation of water quality parameters in aquaculture. It's available in the market. So there's another product which is available in the market where the nanotechnology is there, which people are using in the aquaculture, is the Hydroplex Nano. It's a broad spectrum drip treatment for this parasite. Against the parasites, we can use there are different types of parasites in the fish south in Kashmir. Also, we have this parasite problem with the fishes. So there's another commercial product which is available for the aquaculture, which is a fisheries product. Again, there's Nano 3600. It's a microalgae which they have made in the form of this nanoparticles, and these microalgae are used for the larva culture. It has been as a food for the various larvae in the hatcheries. So this is also available in the commercial level. We can purchase it anywhere in the market. And there's another products which is again anti-parasite, which against the parasites, which is also available. Actually, there is a database where you can find out the products, what the products may be available in the market with respect to this, whether it's a medical, whether it's a textile, whether it's a veterinary and fishery science or agriculture. There's a database. That database name is the Nanotechnology Public Database, where you can find out what are the products, commercial products which are available at the market level, which companies they are manufactured, which country has been that has been developed. So, and then if we talk about the nanotechnology, it's not only the fisheries where it is application. Its applications everywhere. It's everywhere. You can see the particularly this cosmetic and personal care, 60% is contributing everywhere. Whether it's a medical science, whether it's aerotronics, whether it's agriculture, everywhere this nanotechnology nowadays everywhere in our life. A lot of products are available in the market which are related with this nanotechnology. So I want to give you the message before winding up my presentations that Father of Nanotechnology has told us a long back ago, nobody's ever figured out what life is all about. It does not matter. Explore the world. Nearly everything is really interesting if you go deep, and it is if you go anything you go deep enough, you will 100% learn more about that technology and about that techniques. So there's a product, a nano silver toothpaste. So keep using that. Keep smile. It's good. It will protect you from antifungal and antibacterial. It's available in the market. And the slides are also earlier. I have seen other people have shared this slide also. That is the talk of our earlier president, Dr. APJ Abdul Kalam. He told this nanotechnology is growing like anything. It is knocking our door. It's booming. It's incredible. So in India also there are a lot of projects, a lot of institutes which are working. There's a nano mission separately for nanotechnology. And sure, future, whatever I have shown my work, my thing there, that was from the Center Institute. From last year, I have joined this Faculty of Fisheries. Inshallah, and soon we will work on this nanotechnology here also. Thank you very much for your kind attention. If you have any questions, please ask me. Thank you.
Thank you very much, sir, for such an insightful lecture. We'll wait for a few minutes. We'll see if any of the participants have any questions. Yeah, we have one question in the Q&A section. Kindly check.
1 minute.
Yeah, sure.
Any collaboration with? He's asking something. Any collaboration with? Yeah, sure. We are available for the collaboration. Yeah, sure. International, no issue in that. No issue.
All right, Dr. Mohammad Ashraf Rathar, sir, thank you very much. You talked in depth about topics like marine aquaculture, Indian fisheries, nano revolution, and nano aquaculture. We thank you for such a constructive lecture. Thank you very much.
Oh, thank you. It was my pleasure. Thank you. Please pay my regards to all the team, all the congratulations once again for you for organizing Yes, thank you. Thank you very much. Thank you.
Thank you, sir. All right, participants, we are being joined by Dr. Shifa Munir, who has done her PhD in entomology under the able guidance of Professor F.A. Zaki, sir, who is former Dean of Faculty of Horticulture, Shalimar, Coast, Kashmir. And the title of her thesis was Synthesis and Study of Efficacy of Nanopesticides to the Pests of Horticultural Crops. Dr. Shifa Munir, we welcome you in this webinar, and you may kindly proceed now.
Yeah, thank you.
Thank you, ma'am.
Yeah, so Assalamu alaikum and a very good morning to everybody who's listening out to me, to all the participants and the revered organizers. I would just like to know that if I'm audible.
Yeah, ma'am. Yeah, ma'am. You're
Okay,
quite
fine.
audible. Thank you.
Okay, so thank you so much for this opportunity. I'm extremely grateful for this opportunity of presenting my work here on this platform, and I congratulate Division of Pathology, SCOST Kashmir, for organizing this event, this very beautiful, insightful event. And I have been through almost every session, and every session has been really, really very much enriching and informative. So congratulations for that. So we don't have much time, so I'll straight away go to the presentation and the work that I've done, as rightly pointed out by Noreen. I think, yeah, it was done under the supervision of Dr. Farooq Ahmed Zaki. So under the mentorship of Dr. Zaki, I have a heartfelt gratitude for him because this work was something that was very extensive, that was very, what should I say? It was a breakthrough for entomology. It was a breakthrough work, and it was a collaborative approach between different institutes. And nano, as such, we don't have much of nano as of now in Kashmir overall. Maybe we have some work, but not particularly in agriculture or horticulture. So, alhamdulillah, we were able to just get some very good results and do some quality research. So I just extend my sincere and heartfelt gratitude to my supervisor, Dr. Farooq Ahmed Zaki. So let's just get started with the presentation. I'll first share my screen and yeah. Okay, yeah. Okay, so I think it is visible to everybody.
Yes, ma'am.
Yeah. Okay. My work was quite extensive. It was a lot of work. So, but I won't be presenting everything here because we're short of time. So I'll just go through the main things. I'll just give you a crux about the presentation, what basically we did. So I'll try to make it as short as possible. And I have squeezed the presentation to almost 1/4 or 1/3 of the part of what it actually was. So just giving you a quick insight of what basically we did. So yeah, the topic is synthesis and study of efficacy of nanopesticides to the pests of horticultural crops. So basically we synthesized some nanopesticides and then we used those nanopesticides against pests. And then we also compared these nanopesticides with the commercial formulations. So that's what basically we did. So you all know by now that what nanopesticides are, so I don't need to get into the details. It increases the the solubility is increased, there is slow release and all. So that is what nanopesticides are. You know about it, I think. So yeah. So here you can see this. These are what we aim to do. These are the objectives that we aim to do. So first of all, we standardized a technique for synthesizing nanoformulations for 3 pesticides that were thiacloprid, pyromethylene, deltamethrin, and finally we did the characterization. So what is characterization? It's simply to know that your synthesized material falls in the nano range. That is what characterization is. Then after this pesticides were synthesized, after these nanopesticides were synthesized, we went for the bioefficacy part. So in bioefficacy part, we tested the nanopesticides against 3 pests. That is the aphids, the European red mite, and the whitefly. And these 3 pests are quite devastating in temperate crops, as well as the whitefly. This is an important crop in the ornamental crops as well as the greenhouse. So later, what we did is that we also found out some suitable dose for the nano formulation pesticides, because we still don't know that what should be the dose at which we should be spraying our nano formulations. Because obviously, if nano formulation is more toxic, you should be able to know. You don't have to spray it at the recommended concentration at what you use to spray the commercial formulation. You obviously need to decrease that dose. So what that dose should be, we should have at least a rough idea that how many times it is potent, how many times it is to be reduced to reach the level of efficacy that a commercial formulation gives at a recommended dose. So that is what we tried to establish in this objective. And also we studied the persistent residual toxicity, the LT50 values, that is the lethal time 50, and also the rapidity of killing test orthopod pests. And finally, we went for the ovicidal activity, that is, if these nanopesticides were able to kill the eggs of the European red mite or not. So these were the objectives that we tried to, accomplish. So for the first objective, where we try to synthesize the nanopesticides and do their characterization, as I told you, these 3 pesticides, that's thiacloprid, pyromethylene, and deltamethrin. These are the 3 pesticides, and these are the details of the pesticides. I don't need to get into that. So yeah, mite, you all must be almost knowing, I think most of you must be from agriculture background. If not, then European red mite, this is a very, very devastating pest. This has assumed the status of a major pest in apple growing countries. So, and this is again of pathological importance as well, because this, the feeding sites of these mites, when it punctures the leaves, Alternaria fungi just invades those feeding sites and establishes the disease. So when we control European red mite, definitely we are to a large extent controlling Alternaria as well. So this is the pest that we firstly targeted, and followed by green apple aphid. This is again one of the very nasty and a nuisance pest. You can see this pest accumulated on, they are there in a lot of numbers at the tender stems and twigs. You can see it there. So this is green apple aphid, and then these are the greenhouse whiteflies. You can mostly find these whiteflies in ornamental pests and ornamental crops and vegetable crops in greenhouses, because mostly they flourish in higher temperatures. And obviously in greenhouses we have high temperature. Mostly all insects flourish in high temperatures because they reproduce rapidly, they grow rapidly. So whenever we have higher temperatures, you can see that we have a lot of generations of insect pests in that season. So greenhouse whiteflies, these are. So yeah, these were the insect pests, and these were the 3 pesticides that we used to form to make our nanopesticides. So how did we make the nanopesticides now? If you must have gone through Dr. Najm Shakil from IARI, he gave a presentation. You must have just gone through that presentation. So that was a beautiful presentation, a beautiful work, and literally a very brilliant piece of work, because they have they have designed that technique of synthesizing an amphiphilic polymer and then encapsulating the pesticide in it. So that is the same thing that I followed, because this work, part of my research, was done in IARI only, Indian Agriculture Research Institute, under his supervision only. So what we did was that we synthesized an amphiphilic polymer. I don't need to again go into the details, because if you must have followed that presentation, you must be knowing that what this is all about. We synthesized, and anybody having any query, if I'm skipping some part, or because I'm telling you that there's a time constraint, so I won't be just getting into the details. But if anybody has any query and or any clarification is needed, you can just mail on my, drop me a mail, so we can talk about that later. So here you can see that we have synthesized the amphiphilic polymer through polyethylene glycol 600, which is eco-friendly, which is there's no problem with that, because this is used as a laxative also in many cosmetics, in many things. So there's no problem with that. And dimethyl 5-hydroxyisothiolate, this is a linker molecule. So we make a polymer in short, and that too an amphiphilic polymer, that is which has both hydrophobic and hydrophilic end. Finally, when we get the polymer, we don't have the nanopesticide as yet. Then what we do is that we encapsulate the pesticide in the amphiphilic polymers, or in the nanospheres that we've got. So what we did then was that the technique then employed was that we standardized the technique, that how much of ratio of a pesticide or the active ingredient of the pesticide is needed to be put into that amphiphilic polymer, so that it gets encapsulated in that amphiphilic polymer. So this is how we standardize the technique. And this is not so easy that you will just take the active ingredient and put it in the amphiphilic polymer. No, you have to first standardize it. That is, you have to first just find out that what works the best, what ratio of the active ingredient and the amphiphilic polymer works the best, and then only you can just proceed further. So that is how we did it. And we encapsulated the solution finally after, as I already told you, Dr. Najim Shakil has talked about it, so let's not get there. And finally we got our nanopesticide. It was ready. And then okay, so these are just the pictures that I was working in IRA. Okay, so yeah, once our this nanopesticide was ready, what we did was that we characterized it. So these were the nanopesticides that we made. Yeah, so characterization part, as I already told you, is simply knowing that your that the formulation that you have prepared is in nano range. Simply, that is characterization, or just finding out its properties and all. So in simple terms, it's just knowing that your solution is in nano range. Now, these are the available characterization techniques, but for me, what we went for two techniques. That is the transmission electron microscopy, and I'm sorry, but this arrow is at the wrong place. It's actually the dynamic light scattering and transmission electron microscopy, that is TEM and DLS. We went for these two techniques for characterization. So this part was done in Punjab Agriculture University in CIFET, and also in the lab of nanotechnology there in Punjab. So it was done there. They have these facilities there. So I went there with my samples and got it done over there only. So yeah. Okay, so this is the result of DLS. You can see that this is nanothiacloprid graph. This is the graph that you obtain when you work in on a DLS. When you put your solution or sample in a DLS, this is the graph that you obtain. This was a Malvern zeta sizer, and the average particle size was 87.24. These are the D values, and this is the polydispersity index. Next, this was for nanospiromesophin. Again, you can see the average particle size is 40.10, and this was nanodeltamethrin, 68.72 was the average particle size. So this is the specification of the instrument that we were using. So overall, this was the result that we obtained. This is the average particle size, the D values, and the PDI. So what are D values? It is the diameter range. That is, for example, for nanospiromesophin, if we say D90 is 55, this means that 90% of the particles lie well below the 90 nanometer range. So similarly, for D10, we can say for nanospiromesophin that 10% of the particles of the nano solution lie below 20 nanometer range. So this is how we interpret it. And this is the PDI, that is the polydispersity index, how dispersed, how much dispersity is there in the solution. Okay, so okay, so this was the TEM analysis, transmission electron microscopy. Again, this is the TEM instrument. This instrument is the TEM, and obviously you all must be knowing about it. So this is where I did my TEM analysis, and these are the images for nanothiacloprid, for nanospiromesophin, for nanodeltamethrin. And all this was done, as I told you, in the electron microscopy and nanoscience lab in PAU. So this was then analyzed by ImageJ software, a very easy and user-friendly software. You can just analyze your TEM pictures there. So here you can see the in the micelle, the it has engulfed or just trapped the active ingredient of the pesticide within. So this is how it is. So for the objective one, we just found out that we prepared the nano formulation through PEG-based amphiphilic polymers. We characterized it through TEM and DLS. The average particle size was well below the 100 nanometer range, and spherical shape and nano size of formulations was confirmed by TEM. And again, the encapsulation was also confirmed by TEM. So for the second part of the research, we did the bioefficacy of these nano formulations. So we just planted these pots, and a lot of pots were planted. So then what we did, these were the treatments. We took the the nano formulations of, and again the commercial formulation, because obviously we need to compare the nano and the commercial formulations. And for each chemical, we took 5 concentrations. So again, the control was there for water and PEG for nano formulations. And then we simply what is bioassay? So it is just, you just take the leaves of the plants that you have planted. Then you dip it into the chemical, because we had these 5 concentrations through serial dilution. We made these concentrations. We got these concentrations ready in the laboratory, and then we dipped the leaves. We dried the samples. We, the typical bioassay thing that we do. Then we just put the cotton ring around it, and we introduced mites or whatever the insect pest was, aphids and all. And then we put it into the BOD, and then mortality count was done after 24 hours. So in case of whitefly, you cannot just introduce it through a brush. You have to blow it into the aspirator, because whiteflies have got wings, and obviously you cannot control their flying. So obviously you cannot take it through a brush. You have to blow it through an aspirator. So this was done in different nurseries of Kashmir. Finally, what we just obtained, the results that we obtained for aphispomi, that is for aphids, you can see nanothiacloprid has LC50 value of 0.0020. That is very, very less, which means this is the most toxic. The lower the value of LC50, the more toxic the chemical is. And the higher the LC50 value, the least toxic the chemical is. So here you can see spiromesipin is the least toxic, and nanothiacloprid was the most toxic against aphids. So here nano definitely wins. So in case of European red mite, again you can see nanospiromesipin leading, and commercial formulation of deltamethrin is lagging behind. So again here you can see for Triaelurodes vaporidium, that is the whitefly, nanospiromesipin and nanodeltamethrin work the best, because they have the lowest LC50 values, while deltamethrin's commercial formulation had the highest LC50 value, and which is why it is the least potent. So then we just worked out these relative toxicity and relative toxicity folds and all. So this is overall the comparison table. So here we compared the LC50 values. So the summary is simply that everywhere, in case of aphispomi, in case of Avarnicus ulmi or whitefly, we had the nano formulation with the lowest LC50 value. That is the most potent of all, which means finally that acute toxicity of nanopesticides was found to be far more superior than their corresponding commercial formulations. Then what we did determine, or we found out some suitable pesticide doses against each test orthopod pest. Now, how we did that? So as I told you, we cannot use nano formulations at the recommended dose of their counterparts, like commercial formulation. For example, if I'm using spiromesipin at 1%, I cannot use nano formulation of spiromesipin at 1% again, because that will be really very much toxic, because now we find out that nano formulation of spiromesipin is obviously very toxic as compared to its commercial formulation. So what do we need to do? We need to decrease the dose to a level where it, it's at par with the recommended dose. The control is at par with the and it's not toxic. So we tried to work out we tried to roughly work out some of the concentrations where we could achieve good control as that of the recommended concentration of the commercial formulations. So here you can see recommended concentration of commercial formulation. If mean mortality was X, then same concentration for nano formulation was used against pests, and then mean mortality, for example, it was Y. In case this mean mortality, that is for nano formulation, in case it was more than this mean mortality, that is X, then what we did that is we just went for lower concentrations. We went for lower concentrations through geometrical retrogression, and finally we stopped at that point where we got the mean mortality of pests almost equivalent to X. So I'll show this here. Yeah, for example, here thiacloprid recommended concentration is 0.0086. At this, the corrected percent mortality is 60.57. But in case of nanothiacloprid, at this very concentration, the corrected percent mortality is 100. So what we did was that through geometrical retrogression, we went down, 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 and went to the concentration, this concentration, where we found out that yes, 59.81. This is the mortality percentage, which is almost equivalent to our this 60.57. So this is what we call as the baseline concentration. And then we worked out again in this range. We worked out again for we did all the bioassay and all this. This is a very extensive work. It looks like it's not it's very simple, but it is a very extensive work. We went for then this, the range of these concentrations were again used against different pests, and we found out the corrected percent mortality to establish what ranges are working at par with the recommended concentration of the commercial formulation. So we established this for spiromycetin as well and for deltamethrin as well. So now here you can see, yeah, for thiacloprid, concentration 0.0086 is the recommended one. Its corrected percent mortality is this. And for nanothiacloprid, these are the concentration ranges at which the corrected percent mortality is almost equivalent to this. So relative fold change, you can see that 781 to 803% more potency. So that is what this is. So yeah, this is then the average that we took out from the required response limits. So all in all, what we can say is that the dosage of nanothiacloprid could be lowered by 781 to 803% to achieve control caused by recommended dosage of commercial thiacloprid. And same is the case with others. So you can, in short, this means that you can reduce the dosage of a nanopesticide to many, many times so that you can achieve the control, same control caused by the recommended dosage of the commercial formulation of that very pesticide. So that is what the basic summary about this was. Then finally, fourth objective was that we worked out the persistent residual toxicity, LT50, that's the lethal type 50, and also we are, yes, and also a lethal LT50 for persistence. Yeah. So let's just start. Yeah, again, these are the treatments, these are the concentrations, these are the test insects that you know already. So again, what I did here was that we sprayed the plants, we sprayed the potted plants with the different concentrations. So first experiment was that we wanted to know that how quickly will the pesticides kill 50% of one-time introduced insect population. That is, for example, if I come in the morning, I take a leaf from my potted plant, I put 50 insects on it. I want to know that in how much time will 50%, yeah, that is 25 of these insects be killed by a nano formulation. So that is what we wanted to know. That is called LT50 for rapidity. So here for green apple aphid and ERM, treated leaves of potted apple plants were picked. These were kept in Petri plates. Then 30 adult mites were released. And then what happened, that mortality was checked after 3 hours, 6 hours, 12 hours, 24, 48, 72 hours. So whiteflies, in case of whiteflies, we just blew it into an aspirator because obviously, again, as I already told you, it has got wings, it can fly, so we cannot just control it with a brush. So yeah, in terms of LT50 of rapidity, if you see nanothiacloprid in case of green apple aphid at this 0.0086% concentration, this is very rapid in killing the killing green apple aphid, but nanothiacloprid here lags behind here if it is used at a lower concentration. You can see that the LT50 for rapidity is obtained very, very late. Here it kills 50% of the population at 3 hours only, but here it kills at 35.48. This means at lower concentration, it is not that potent in rapidly killing the pests. But commercial formulation here wins the game. And similarly, in case of spiromycetin, but in case of deltamethrin, it's different. So yeah, that was about rapidity. Then these treated apple leaves were again plucked. This is the second part of the experiment. Then again, we introduced insects on it. And again, it was checked, but this time after 24 hours. So then after 24 hours, when mortality was checked, then new treated leaves were taken the next day, and new batch of insect was put. And again, it was mortality was checked after 24 hours. So this procedure was continued till no mortality or negligible mortality was there. So then we determined the persistent toxicity. That is the method given by Sarup and co-workers. So yeah, here you can see that nanothiacloprid at 0.0086%, it persists for 21 days, while lowest was the thiacloprid. This means here nanothiacloprid at lower concentration persisted more as compared to nanothiacloprid at the higher concentration. In previous thing, we found out that nanothiacloprid was at 0.001085%, it was lagging behind. There was, it didn't kill the pest very fast. But here you can see, though it didn't kill the pest very fast, 50% of the population, it didn't kill very fast. But here it persisted more as compared to the commercial formulation, which means that even at lower concentration, the nano formulation might not be very much potent to rapidly kill the pest, but it can persist longer as compared to the commercial formulation. So that is there. Then again, here you can see the same trend, and for deltamethrin, you can see the same trend. And then the same thing that we did previously, for after 24 hours, we introduced new leaf, new insects and all, but then the analysis part was different. Here we used the probit regression analysis, and that's given by Finney and the Abbott's formula and all. So we did that to find out this LT50, that is persistent of 50% mortality. That is till what day will the pesticide, kill 50% of pests. So that is LT50 persistence. So you can see nanothiacloprid till 11 days, it caused mortality of 50%. In case of nanospiromycin, till 12 days it did so, and in case of nanodeltamethrin, again till 12 days, and followed by, in each case, nanothiacloprid at lower dose. So here again nanospiromycin at lower dose, here again nanodeltamethrin at lower dose. So the commercial formulations again lag behind here. So this is the summary of this thing. So I don't need to go there. And the last objective was the ovicidal action against the eggs of European red mite. Here what we did, we just got the ERM eggs from the orchard. We introduced it on the leaves. We sprayed the eggs with different concentrations of nano as well as commercial pesticides. And then hatching was observed in the control, because once in the control there was only water or peg. So when the eggs hatched from the control, this was the time that it should be hatching from the treated leaf as well. And so we just found out whether the eggs are hatching from the treated leaves or not. So similarly, we just recorded the data and we got the percent egg hatching. This we just computed, and percent egg hatching inhibition. Again, we computed these two things. And what we found out was that nanospiromycin was the most potent in controlling the hatching of eggs of European red mite. And the least potent was deltamethrin's commercial formulation, which was lagging behind, which was the last in the league. So yeah, this is how we computed it. So this is very important because, though nano formulations aren't commercialized yet, they're not, we don't have any permission to spray them in the field. We don't have any guidelines. But later, if someday in future it happens, what nanospiromycin actually we using spiromycin we are using in our orchards, against eggs or adults of European red mite. So in future, what we can do is that when we spray spray spiromycin once, we'll spray nanospiromycin once in the orchards, it'll control eggs. But in the spring, we won't be needing one more spray for adults of European red mite, because it'll take care of the eggs and it'll take care of the adults as well, because it's quite potent against both. So this is, this is where this is the application of nanotechnology, that there will be reduced rounds of sprays. So that is very important here. So this was the summary. So finally, what we can claim from our research and with what we were able to do was that we increased the efficacy of the pesticides, durability, and reduction in the amounts of active ingredients that need to be used, enhanced apparent solubility, controlled release, targeted delivery, protection against premature degradation, enhanced uptake and efficacy, enhanced toxicity to target organism, that is lower dose, less rounds of sprays, and allow the active ingredient to be released in the pest environment in a slow and targeted manner, improved operator safety, increased apparent solubility of poor water-soluble active ingredients, which result in improved tag mixing, better cuticle penetration, and uptake. So yeah, these are all the claims that we can make through our research, and we have, we have all the data, all the analysis, and all the proofs for all these claims. And nano formulations do work really well, at least in the laboratory conditions. They work well. We cannot go for the field trials though. So nanotechnology applications in agriculture is an emerging area, mainly because of dose reduction and high efficacy, but requires extensive research and techniques in agriculture pest management sector. So yeah, so this study was carried out in the experimental area at Division of Entomology. Really thankful to the Division of Entomology. And without the support of these very institutes, IARI, PAU, CIFET Ludhiana, then I was selected for the Nano Mission School of Institute of Nanoscience and Technology, Mohali. So thankful to that as well. And NABI, that is in Mohali, Punjab, then SIAB in Mohali, Punjab, NIT Kashmir, IIT Kharagpur, and Nanotechnology Congress, Dubai. I'm really thankful to all, and also thankful to the National Inspire Fellowship from the Department of Technology that is duly acknowledged. So thank you so much, everybody, for patient hearing. Thank you. That's the presentation that I had for you. So if there are any questions, please let me know.
Thank you, Dr. Shifa. Very nice presentation.
Thank you.
Of course, an exhaustive work carried under the kind guidance of Professor Farooq Ahmad Zakki sir, yes, our ex-dean Faculty of Horticulture. I think you can take the questions from Q&A. You please check it on your own. Sure. I think only one question is there.
Yeah. So yeah, safety to beneficial insects. So, Dr. Ansar Ali, we haven't actually tested the safety to beneficial insects. We haven't done any work about that. But yes, I can tell you one thing, that when we spray, normally when we just spray pesticides, obviously those are also toxic to the beneficial insects. But here again, we are just manipulating the size, and we are using PEG. PEG is totally eco-friendly. There's no harm that can be caused by PEG, because even humans take PEG. So there's no harm that this PEG can cause. So not an issue with that. But yeah, about the size, there can be a deeper penetration in the beneficial insects as well. So we just need to, spray it according to the, for example, when we know that this is the time when ladybird beetles will emerge, we just need to skip that time, and we just need to avoid spraying our pesticides during that time. But that is common for nano formulations, and both, the formulations that we are using this time also. So that remains a common factor for both. Okay, Tanzila is asking, sorry, what's the possibility to go for field trials? I mean, when it can reach farmers? Okay, so I don't think there's any hope of testing it in fields, because we still have no guidelines, and this is not allowed to be sprayed in the fields. But yes, what you can do is that you can go for the pot culture like I did, and that's almost equivalent, almost, not actually, not very much, but almost equivalent to field trials, because the pots are there outside in the environment. So you can go for pot culture. And about farmers, I would say that as of now, we don't have any guidelines, and we cannot recommend this to farmers. So yeah. Okay, compatibility with other. Sorry, but I'm not able to understand what compatibility.
Maybe compatibility with the fungicides and others, but these are all research trials going on. Nothing forward has.
Yes. We didn't study that. I'm sorry, I cannot just answer this question for you, because we didn't go for that.
I would like to read some comments of our participants for you. Huh?
Sure.
Excellent presentation by Shifa. One of the participants has written excellent and dynamic presentation, and presenter, and others, mashallah, and other things from different our participants. Thank you for responding to our short call, and for a nice elaborative. Of course, it was your research work. It gives it's immense pride for us that such a type of wonderful work has been carried out. One of our student in SCOST Kashmir Faculty of Horticulture. Thank you, Dr. Shifa. Thank you.
Thank you for having me. Thank you.
Now an announcement for the participants. We will be waiting for just few minutes and we will start our valedictory function. Please bear with us, stay for few minutes, then valedictory function, it will take hardly 20-25 minutes and it will be over after that. But it is compulsory for all the participants to be present during this valedictory function. Please stay with us, bear with us for 5 minutes. Then after we will be starting that.
Do not forget to subscribe our channel by hitting the below subscribe button and press the bell icon.